I want to start off by saying welcome everyone to another MassMedic webinar. We're thrilled to have you here. I'm Rachel Robinson, Chief Operating Officer of MassMedic, and I know you're in for a really excellent program. So before we get started, I would like to remind you that we want to leave lots of time for Q&A at the end. So please feel free to put your questions in the Q&A option at the bottom of your screen. If you send them in through the chat, sometimes they get overlooked or missed. So please use that Q&A option and we'll be sure to answer as many as possible at the end of the program. Additionally, we will be recording this program and providing the recording to you afterwards. The slide will also be made available for you, thanks to our partners at Boston Analytical. So please don't feel free to pay attention and not like you have to take 1,000 notes. You will have access to the recording and presentation afterwards. So with that, I would love to say thank you to Eric and Nicole at Boston Analytical for putting together what I know is going to be a really outstanding program. You know, transdermal drug delivery is something that is clearly a hot topic and on the rise. And it's always interesting to see FDA's reaction and response when something is different and unique and new regulations and guidance come around. I've also always been very fascinated with the idea of simulation studies when it comes to FDA requirements and regulations. So this webinar actually kind of put together all of the things that I'm particularly interested in. So I hope all of you get as much out of it as I know I will. But Eric, I'm going to turn things over to you and let you take it away.
Great, thank you, Rachel. Let me share my screen here. Rachel, are you able to see that?
Looks great.
Great, thank you. All right, well, good morning to everybody. Thank you for your attendance this morning. My name is Eric Hill. I'm a Senior Director in charge of the Chemistry Laboratories at Boston Analytical. And today I have the pleasure of speaking to you about extractable leachables testing of transdermal patch products. So today I'm going to start off with just an overview of what a transdermal patch looks like. There's a couple different types. Then I'll go into extractable leachable regulations, particularly in the US for patch products. Then go through a general workflow for evaluation of extractable leachables of patch products. This will include materials risk assessment, extractable studies, sweat simulation studies, leachables method development, and leachables validation study plans. And then I'll finish up with a case study. And as Rachel mentioned, we'll have time for some Q&A after all of that. So starting off with just a general overview of patches, just to kind of set the stage for everybody. There's two types of drug delivery patches. The first one is called a drug and adhesive transdermal patch. And this is the simpler of the two forms. It's really got three main parts. The first is what's called a backing layer. This is really the meat of the patch, provides the physical structure of the patch. It's usually made of a non-woven material, often a polyethylene terephthalate. There's also a release liner. This is what protects the patient contact surface prior to use. And so anybody who's ever come in contact with a patch before, there's usually a liner that you peel away and discard. That's the release liner. And then there's the adhesive matrix. And this is a gel type material for this type of patch. It's typically contains a polymer material, and it serves really two purposes. One is it's the adhesive that allows the patch to stick to the dermal surface of the patient, but it also contains the drug mixed into that adhesive matrix. And so the drug is administered and diffuses out of that adhesive and through the dermal layer, during use. And then all of that is packaged usually in a pouch type of package. This is a sealed, sterile, protective pouch that holds the patch prior to use and is usually torn open at application. The second type of transdermal patch is called a reservoir patch. And this one's a little bit more complex than the other patch. It's got a few additional layers, but really the main difference is it has a reservoir, so that the drug product is contained in this reservoir, and that's where that drug is contained. It's usually a semi-liquid form, but similar to the other patch, there's a backing layer. This again provides that physical structure for the patch. There's the drug reservoir, as I mentioned. This is an open reservoir that holds that drug product for release. There's also a drug release membrane, and this physically holds that drug in the reservoir. It's a woven material, but it is semi-permeable and will allow that drug to release through into the dermal surface. There's also a contact adhesive. that while that adhesive doesn't contain the drug product similar as the other patch does, it does provide the same benefit that allows the patch to stick to the dermal layer and stay on that dermal layer during use. And then this will also be sealed in a pouch similar to the other patch. And so if we look at these, from a cross-sectional layer, you can see again that the matrix patch is on the right. It's a simpler patch overall in terms of number of layers and number of materials. It's got that release liner, which will be peeled away, discarded, and then you've got the drug and adhesive layer and the backing layer, which provides the backbone of the patch. The reservoir patch on the other side, on the left side, has got additional materials that are involved. Again, it's got that release liner that's very simple or very similar to the other patch release liner, but then it has a contact adhesive, which serves just the sole purpose of allowing adhesion of the patch to the skin surface during use. It's got a rate controlling membrane. Then it's got that reservoir that contains the drug product. And then again, the backing layer, which is the backbone of the patch. So now getting into regulations for specifically extractable leachables regulations for drug delivery patches. We start out with the 1999 FDA container closure guidance. This table comes directly from that guidance. This table is where we look at drug product forms along two axes. One is the likelihood of packaging component and dosage form interaction. So how likely is it that there's going to be an interaction between the packaging and the dosage form, and that interaction could cause leachables to form? And then on the left axis of this table, you can see what is the degree of concern associated with the route of administration. And so when we talk about route of administration, that's the route that the drug is going to be administered to the patient. And so if in degree of risk for different drug forms in this table really starts from the upper left, those are the high or highest risk drug forms, and then it goes down in risk to the bottom right, which should be the lowest form of risk for these drug forms. And so if we look at the bottom right, we can see oral tablets, oral capsules. These are the lowest risk in terms of leachables. And that's because really our GI tract is very good at dispelling toxins that may enter our body. We may get momentarily sick if we have toxins introduced to our gastrointestinal tract, but overall, it's pretty adept at dispelling those toxins and preventing them from being absorbed into the bloodstream. However, if we go up to all the way to the highest risk drug form, which are inhalation aerosols and sprays, if we inhale a toxin, generally that's going to be absorbed in the lungs. It's going to go right into the bloodstream, and then there's risk of toxicity to the patient or to the body. And if we talk about transdermal ointments and patches, I've got that circled there in red. These really, while they don't have the highest form of concern with route of administration as the inhalation aerosols do, they still have a very high concern, and they also have a very high concern in terms of likelihood of packaging component and dosage form interaction. So, what the takeaway here is with regards to transdermal ointments and patches, we know we've got high concern for leachables, not only forming, but also a high concern that those leachables can be risk to the patient during administration. And so leachables are something that we've got to be concerned about, and we've got to make sure that we design an adequate study to monitor those leachables and ensure that we've got a safe drug delivery system. In terms of extractable leachables testing, particularly in the US, we really start out with USP. And the USP has got a myriad of chapters which govern extractable leachables testing. And they're really designed to be an all-encompassing kind of worldview in terms of extractable leachables, starting with USP 661.1, which is compositional information of packaging materials. So in this testing, it's a lot of kind of more basic chemistry where we're looking at ID of material, we're looking at physical properties, and this is where we're testing materials that are going to go into packaging. From there, we would graduate to USP 661.2, which is packaging systems. This is some additional physical testing and ID testing of actual formed packages. And then at the conclusion of that chapter, we get into USP 1663, which is extractables testing. And that chapter guides us in doing extractables studies and extractables testing of packaging systems. And then from there, we move into USP 1664. This is leachables testing. By definition, leachables testing is always done on drug products. So again, extractables is packaging systems, leachables is drug products. And then there's also USP 665, which is processing systems. That really applies more to drug product manufacture. In this case, we're really talking about a drug delivery patch. Another set of regulations that we have to also think about is ISO 10993. And ISO 10993 is a set of regulations that includes extractable testing, as well as biocompatibility and some other testing for medical devices. And a drug delivery patch really kind of blurs the line between a drug product and a medical device. And so we've got to be cognizant of both those regulations. And so when I get into the workflow, we'll see a little flavor of both of those. So again, when we just an overview of extractable leachables testing, when we talk about extractable studies, our goals here really are to predict potential leachables at the end of shelf life, so worst case scenario. And any data that we generate is going to be correlated with a future leachable study. The data is used for preliminary tox risk assessment. It can be, doesn't always have to be. It'll provide a detailed extractables profile for each container closure system component. And that extractables profile will take the form of looking at organic volatiles, organic semi-volatiles, organic non-volatiles, and inorganics. And it involves extraction of the packaging system components with multiple solvents with temperature and analysis using those multiple techniques to analyze for those different forms of extractables. So, you know, kind of in a nutshell, when we're talking about extractables testing, we're talking about using solvent extraction of the packaging system materials themselves. We're not doing anything with a drug product when we do extractable studies. Conversely, when we get into leachables testing, this is where we're looking to monitor leachables in the drug product over the shelf life of the product. And again, we're always looking to correlate data between extractable studies and leachable studies and back and forth. We also want to have a goal of determining maximum leachables levels throughout the product shelf life. And this will also use this data to establish specifications and acceptance criteria for those leachables levels. And again, just in summary, leachables testing, always done in the drug product. So we start with extractable studies. We extract container closure system materials, get an idea of what that extractables profile, what the potential leachables are in those packaging materials. From there, we move into leachables, and we monitor leachables coming from packaging, but we monitor them in the drug product, what actually is leaching. And we use the information from extractable studies to tell us what to monitor for. That gives us an overview of what potentially is there and could leach. Now, specifically when we talk about patch regulations, as we talked about, transdermal patch products are high-risk drug forms. They're going to require full assessment of extractable leachables. If we were talking about a low-risk drug form, such as a solid oral dose tablet, you know, we'd certainly look extractables, but we probably would do some minimal leachables testing. But again, with a transdermal patch being such a high-risk drug form, we're going to need to do a full assessment and monitor extractables and leachables throughout that shelf life. With our extractable study, we're going to make sure we design those very carefully, such that they generate worst-case extractables profiles for all the materials incorporated in the patch. And I reviewed those previously for the two different patch forms. And then our leachable studies are going to need to be performed such that we monitor those leachables throughout the entire shelf life of the patch, and that we monitor those at various time points, similar to a stability study throughout that shelf life of that patch, whether that's 2 years, 3 years, 4 years, 1 year, you know, whatever that shelf life is. And we want to make sure we monitor those leachables under the use conditions of the patch, including sweat simulation studies, which again I'll define and get into in much detail in a little bit. Now, what are the FDA expectations for patches? And you know, just to pause a minute on FDA expectations when it comes to extractable leachables testing. For those of you that are in the industry and have had any FDA interaction with regards to extractable leachables, you've probably noticed that the FDA expectations of extractable leachables have really changed over the last 6 months, year, 2 years, 5 years. They've changed dramatically. And for the most part, this has really been industry driven. As we've learned more about extractables and leachables from packaging of drug products, and as we've incorporated more and more different forms of materials that we use and encompass our container closure systems, and thus come in contact with our drug products, they present unique challenges with regards to extractable leachables, and those challenges really need to be met through testing, both direct chemical testing as well as biocompatibility testing. And because we've got those unique challenges, and there's so many different forms of container closure systems and drug forms on the market, the FDA is really kind of ramped up and tightened the screws on their scrutiny to ensure that extractable leachables are thoroughly monitored and tested for, to ensure, again, that we've got a safe drug form that's free of toxic leachables that is administered to the patient. And again, with regards to this being industry driven, there's several industry meetings similar to MassMedic, the meeting that we're in now, where there's discussion and data presented from laboratories and from drug product manufacturers on extractable leachables. And the industry is really leading the charge to making sure that, again, we've got adequate and appropriate testing of extractable leachables such that we have safe drug forms. So, but specifically for patch products, here's some comments that we've seen at Boston Analytical in IND meetings of our clients. This is some of these are actually specific quotes. First one, evaluation of extractable leachables from the drug container closure system or device should include specific assessment for residual monomers, solvents, polymerizers, et cetera. So this is the FDA saying that, when we do our extractable leachables evaluation of those container closure system, we got to make sure that we specifically target the residual monomers, solvents, polymerizers that we're going to expect from those materials. So it's not, we got to make sure we design our studies not just blindly following a basic set of rules, but we evaluate the materials that are in our system, and we specifically look not only for things that we wouldn't expect, but we specifically look for what we would expect. So if we're analyzing a PET, we want to make sure we look for monomers and solvents, polymerizers that are specific to a PET material. And then for all drug products, we need to make sure we establish our analytical evaluation threshold, or AET, to be able to detect potentially carcinogenic and genotoxic compounds per ICHM-7 qualification thresholds. So for genotoxic, this is not more than 1.5 micrograms per day. And then from a general toxicity perspective, the AET must be able to detect and identify any leachable that is present in the product at 5 micrograms per day or higher. And that 5 micrograms per day or higher limit comes specifically from ICH and other industry guidance regarding dermal application. For a dermal application, general toxicity, the limit is 5 micrograms per day. If we're talking about genotoxic compounds and partially carcinogenic, that limit goes down to 1.5 micrograms a day and can even go down to 0.15. However, that's usually reserved for inhalation products. So now transitioning from regulations into a general workflow for a transdermal patch product. So the first thing that at Boston Analytical we always recommend is to do a materials risk assessment. And this is an assessment of leachables risk prior to design and execution of the extractable study. And a materials risk assessment is not something that's required by the agency, but what it does is it allows us to take a kind of a global look at all of the materials in the packaging system, all materials as part of the patch, the drug product chemistry, and start to design our study specifically for those materials, as I mentioned previously from the FDA expectations. And it allows us to take a risk-based approach. And a risk-based approach is that specific language from the FDA that they've been mentioning for multiple years now when it comes to extractable leachables. And so the materials risk assessment really allows us to set the stage for that risk-based approach. And so in particular with a patch product, we're going to review all those materials associated with it. The backing liner, the release liner, the storage pouch, the adhesive matrix. We're going to evaluate those materials and start to document what types of materials are they, what sorts of residual monomers, polymerizers, residual solvents, other chemicals that could be present in those materials. Let's evaluate and document those, so we make sure that when we design our methods and our studies going forward, we design them specifically to look for those, as well as other things that could be unexpected. And then the output of this materials risk assessment is that we'll generate a study plan and a formal risk assessment report that, you know, can be used for regulatory review. We've had these risk assessments actually go into the FDA and be reviewed by the FDA and reviewed, you know, and been received favorably in terms of, again, taking that risk-based approach. Once we complete that, then we're going to dive into our extractable study. So if we talk about a drug in matrix patch, our test articles are going to be that backing liner, many of which are manufactured from PET. Oftentimes it'll be printed, so we've got to think about that printing ink. There'll be the release liner, and then there'll be the aluminum pouch that is used to package that. For a reservoir patch, we've got the PET backing liner. We've also got a release liner. We've got the contact adhesive. We've got that semi-permeable membrane. And then again, we've got the aluminum pouch. Once we documented what sort of patch we have and what the materials are and what the components are, we're going to move into what's called a controlled extraction study. And this is defined very specifically in USP 1663. So each component material is going to be extracted with neat solvents. That's going to generate a worst case extractables profile. And some, you know, example extraction media that we may employ, depending on the chemistry that is involved, could be water, 10% IPA, 90% PBS solvent, and then dichloromethane. Those three solvents give us a range of chemistries, range of polarity, so we can get a full extractables profile of those materials. And then typical extraction conditions could be 55 degrees C for 72 hours. Usually this is extracted under maceration conditions. Maceration generally means soaking conditions. And we would advocate for duplicate or triplicate preparation of one lot. Also, when an extractable study is designed, oftentimes you'll do what's called a simulation study. That's defined specifically in USP 1664. But with regards to a patch product, we're going to look at doing what's called a sweat simulation study. And so here we're doing an extractable study, again, where we're extracting essentially the components of the patch. But the extractable study is very focused, and it's going to mimic the use conditions of the patch. And that's going to include not only time and temperature, but also the chemistry involved. And so we're going to extract with a sweat simulating solvent. And there's certainly several in the literature available. One that we've used before in the past is a 10% IPA, 90% PBS buffer adjusted to a pH 4.5. But again, in the literature, there are several other simulated solvents, sweat simulating solvents that are available for use that can be documented. And in this case, you usually would extract at 40 degrees C. That's going to, again, mimic temperatures that, you know, you'd see at the dermal layer during exercise use or, you know, extreme use when sweat is being formed. And we're going to want to try to target a single-sided extraction, because again, this is a sweat simulation study. So in the controlled extraction study, we're trying to target extracting the individual materials so we get individual extractables profiles of each of the materials in the patch. In this case, we want an overall profile of the patch that simulates use. And so if we can, we want to try to do what's called a single-sided extraction. We want to only extract the side of the patch that contacts the dermal layer, not the outside of the patch that doesn't really have direct contact with the dermal layer. And so at Boston Analytical, we've acquired these extraction vessels that you see here in this picture. I'm not sure if you can see my mouse going here. Actually, I believe I can draw on this. Let me see if this will work. Okay, so in here, kind of what you're seeing in red here, this would be where we would put that sweat simulating solvent. We'd fill the vessel there. And then over here, you can see a glass, kind of almost looks like a watch glass, far to the right. What we would do there is we would take the patch and we would stick the patch on that watch glass with the adhesive facing out. And then that watch glass would get clamped on the end of this vessel here, such that we then can perform an extraction where the solvent in here is only going to contact the adhesive side of the patch. And so we get that single-sided extraction. And then we would usually do that extraction again under 40 degrees C temperature for the maximum use time of the patch. And so if the patch that's adhered for a maximum of an hour a day, we'd extract for an hour, again under 40 degrees C, which mimics that exercise temperature that the patch is going to see. Just 1 second. Okay. And then from there, once we generate those extracts, whether it's the solvent extracts or the sweat simulated solvent extracts, we're going to go into extract analysis. And the extracts are analyzed for volatile organic compounds, semi-volatile organic compounds, non-volatile organic compounds, and inorganic impurities. We're going to utilize headspace GCMS for the VOCs, liquid injection GCMS for the SVOCs, liquid or LCU VMS for NVOCs, and then ICMS for the inorganic impurities. Data from the controlled extraction study and sweat simulation studies will be evaluated. All compounds above our analytical evaluation threshold will be identified and semi-quantitated. The data will be correlated between the controlled extraction study and the sweat simulation study. So we're going to compare those two extractables profiles. And if needed, we can do some toxicological evaluation of any of the compounds that are detected above the AET. And the output of this extractable study is a final list of what are called leachables targets, or targets of concern. So all of those compounds that are above the AET are going to potentially be a leachable target of concern. Those are going to be compounds that we want to monitor in our leachable study going forward. So more specifically, extractable study looking at a drug and adhesive patch. So our leachables methods need to be developed and validated, as I mentioned, for those extractables targets. And when we go into leachables, we're going to develop and validate specific methods to analyze for those leachables in the drug product. And in the case of a drug and adhesive patch, we've got to develop methods that allow us to analyze that drug and adhesive matrix. So you can imagine that presents a very tough challenge, analyzing an adhesive matrix, which is usually going to have a polymer involved, to specifically look for leachables that have come from the backing membrane, the release liner potentially, the pouch that it's packaged in, or they could even be leachables that come from interactions between those materials and the adhesive. And so we certainly have a challenge in front of us. We talk about a reservoir patch. Here again, if we're analyzing and developing methods for leachables analysis, we're analyzing the drug product, which specifically in this case is the drug product that's in the reservoir. And so we don't have quite the challenge that we have for the other drug product, in that we're not analyzing an adhesive with a drug product mixed into it. Here we've got a clear drug product solution that's in a reservoir. We can sample that very simply and then develop and validate those methods to target the extractables, or the leachables in this case, in that drug product. So when we move into the leachable study, here we're going to take patch samples, we're going to store them, make sure that, again, if they're printed, that the printing is included. They're going to be in their pouches, and we're going to store them under normal storage temperature for the shelf life, as well as under accelerated conditions, very similar to a kind of chemistry stability study. And then we're going to pull those samples at standard ICH stability storage time points. Typically for normal storage, this could be 0, 3, 6, 12, 18, 24 months for a 2-year shelf life batch. And then under accelerated conditions for 1, 3, 6 months, for a 2-year shelf life. And those could be adjusted depending on the shelf life of the patch if it was not 2 years. And then we're going to employ our validated leachables methods to analyze those target leachables, as well as any non-target leachables that may develop in the patches. So that really concludes, talking about patches in general, going through regulations, and then a general workflow. now I'd like to present a specific case study that we had for a patch material or a patch product here at Boston Analytical. So the case study that I'm going to go through here involved a lidocaine transdermal patch. It was a drug and adhesive design, which I mentioned earlier, certainly presents more of a challenge with regards to leachables testing as compared to reservoir patch. The patch was applied to the skin, used for up to 8 hours with exercise. And again, just to review, a transdermal patch, drug and adhesive, has a backing membrane, has that drug and adhesive matrix, and then has the release liner. So this particular case study, there was some interaction with the FDA prior to employing the study, and they did communicate what their expectations were as part of their non-clinical review of the NDA. And they had specifically directed that headspace GCMS analysis of the patch be done for volatiles. As I mentioned in the overview of a typical study, this is something we would always employ, but they specifically called out that be done. They also directed controlled extractable studies using a 0.9% saline solution, 70-30 IPA, 0.9% saline solution, as well as methylene chloride. They also directed specifically that a sweat simulation study be done. And they directed that the simulated sweat be a buffer solution at pH 4.5, and also that it be done at 40 degrees C for 8 hours. Now, the 40 degrees C, as I mentioned before, that mimics use conditions under exercise. And then the 8 hours was directed because the maximum use time for this patch in particular was 8 hours. And then they directed that the leachable study be performed, including the target compounds from the extractables work. They also directed specifically the leachable study be able to target 2,6-xylidine. This is a lidocaine degradant. This was a little bit of a strange comment, honestly, from the FDA, because it's a degradant, and they were directing us to analyze for a known drug product degradant as part of a leachable study. Generally, a degradant would be analyzed as part of degradation testing, drug product degradation testing, or related compound testing, but they specifically mentioned that they wanted that to be included in the leachable study, so we did that. They also wanted to make sure the leachable study included any other lidocaine impurities. Again, a little bit outside the norm for an ENL study, but it was the FDA expectation, so we complied. And then they directed that sufficient data be supplied to demonstrate safety of the adhesive matrix components. And really what they were looking for there is that they wanted us to perform literature-based tox risk assessments of those compounds. And so again, we've got this drug and adhesive patch product, and the FDA was saying they wanted to make sure that we tested sufficiently for not only leachables coming from backing layer, release liner, but also anything that could follow the drug through the dermal layer during administration from that adhesive matrix. So the first thing we did for this study, again, was a materials risk assessment. We looked at the backing liner for this product. It was a non-woven PET. We knew from that PET we could have polymer additives, we could have residual monomers. We knew what those were. We documented those. We also looked at the release liner, also was PET, and we expected that it would have a similar profile as that non-woven backing liner. The storage patch was laminated aluminum. We knew that laminated film could produce monomers, residual solvents, and additives, so we needed to document what those were or what they could be. We also had that aluminum layer, which was protected by the laminated film. And however, that could be a source of potential metal leachables, but also we knew that the aluminum layer is a very good barrier to migration of leachables from outside environment, including ink components. And so because we had that aluminum pouch, we didn't do any studies to look for ink migration from printing on the pouch. If this was in a polymer pouch versus an aluminum pouch, we would be concerned about ink migrating through that polymer layer, but it's been well documented that aluminum provides an excellent barrier to ink migration. So in this case, we didn't have to evaluate that. We also looked at the adhesive matrix. In this case, it was a hydrocarbon-based resin. From that, we knew we would need to look, and we would likely see saturated hydrocarbons, other hydrocarbons. There was also a hydrocarbon gel involved that was plasticized. There was a styrene, isoprene, styrene black copolymer involved. We've got, various monomers and oligomers from that material as well. And so, we had a pretty good idea what to expect, and we documented all that. And so that was very helpful. And then we assessed the leachables risk based on the drug form, which I mentioned as part of that 99 container enclosure guidance. And we designed the study accordingly and included that in our materials risk assessment as part of a study plan. Once we completed that, we really had our plan. It was a matter of getting the lab and executing. And so first thing we did is we did a controlled extraction study. Our test articles were the backing liner printed, the release liner, the aluminum pouch. The AET that we calculated for this study was 2 micrograms per mil. And again, that was based on a 5 micrograms per day threshold, which I explained earlier. Our extraction media were water, 10% IPA, 90% PBS, and dichloromethane. We extracted at, under maceration conditions at 55 degrees C for 72 hours. And we did triplicate preparation of 1 lot. We analyzed the extracts, volatiles by direct headspace GCMS, semi-volatiles by liquid injection GCMS, non-volatiles LCMS, inorganic impurities by ICP-MS. And we analyzed all of the extract solutions, water, IPA, BPS, and dichloromethane. With the exception of, we did not analyze the dichloromethane for volatiles nor for inorganics. And that was really due to the fact that, first of all, we don't expect to have any inorganics being extracted by dichloromethane. And then also dichloromethane is extremely volatile and produces a very large interfering peak in the volatiles analysis and really renders that analysis kind of null and void. Once we executed that analysis, the summary of what we saw, very complex extractables profiles from the dichloromethane extracts. As we would expect, dichloromethane is a very powerful extractant. We had less complex profiles for the water and the IPA PBS extracts. Multiples extractables, however, were detected above the AET. As expected, and as we documented, we saw hydrocarbon resin components, including alpha-pinene, terpenine. We also saw styrene, isoprene, black copolymer monomers, several of those. We saw the 2,6-iodine impurity from lidocaine, as the FDA directed us to look for and as expected. We saw various residual solvents, including toluene, organic acids, and then we saw some lithium and antimony in the inorganic analysis. And this is a table of that inorganic analysis. Again, you can see the antimony as well as the lithium. Other than that, it was very clean. From there, we went into executing the sweat simulation study. As I had reviewed previously, looking at that data, we had very good agreement between the sweat simulation study and the IPA PBS buffer extracts from the controlled extraction study. And that's really important that we compare those profiles and we verify that we have good agreement. If we had 2 profiles that were extremely different between our sweat simulation study and our controlled extraction study, that would tell us that one of those studies was not designed appropriately, and we need to go back to the drawing board and repeat some analysis and potentially change our plan. But in this case, we had very good agreement. Essentially, the sweat simulation study was a subset of the controlled extraction study data, again, which is what we would expect. The controlled extraction study is designed specifically to be worst case, whereas the sweat simulation study is really mimicking worst case use, but not necessarily worst case extraction. And so we expect that to be a subset, and it was. We did not see the antimony and lithium. We also saw, less components, as I mentioned, from that adhesive matrix. The 2,6-iodine was observed, and overall, again, it was a subset, as I mentioned. We look at the inorganic analysis. Again, the sweat simulation study data was very clean. We did not see the lithium or the antimony, as well as anything else. So once we had all that data assembled, we then went out to a toxicologist and did an assessment of all of that data. And our goal there was to generate a list of targeted leachables for use in leachables method development and validation. As I mentioned, we saw a lot of components. We saw very complex extractables profiles, as expected, because we're analyzing these polymer materials. And so we need to come up with a workable list of target leachables. And so The toxicologist reviewed all of that data, really separated into two categories: red flag compounds and unknown compounds. Red flag compounds were ones that were known to have, more potential for lower limits or lower PDEs would be of concern. And then any compounds that had unknown toxicity. We did an in silico assessment, and then we included those in our compound list going forward. And so from there, we generated our final target compound list based on the following assumptions. First of all, extractables data from the IPA and PBS solvent and the direct headspace analysis were the most representative of, worst case potential leachables. Also, looking at the concentration profiles of our extractables compounds, higher concentration extractables obviously are more likely to become leachables. We also focused on compounds with a molecular weight less than 500. And this is based on a position paper from the OECD regarding dermal absorption, that when it comes to dermal absorption, compounds with molecular weight lower than 500 are most likely to absorb versus ones above that are less likely to absorb. Doesn't mean they won't absorb, but again, when we're talking about coming up with a final target list from a large set of data, we use this assumption to help us to, come up with that focused list. And then we certainly wanted to make sure we chose targets that allowed us to obtain a suitable reference standard. Once we had that final list, then we went into leachables method development. Our initial strategy was to attempt to scrape that drug product adhesive matrix from the backing liner. Again, when we talk about leachables testing, we're always targeting to analyze the actual drug product itself. In this case, the drug product was in that adhesive. So we attempted to scrape that from the backing liner. And we attempted that under room temperature conditions. We refrigerated it. We tried freezing it, even down to -20. Unfortunately, none of them were successful. The adhesive was just too sticky. It was just not really feasible to get a sample off there in any reproducible way. And so we decided to move forward for leachables testing using the scheme that we developed for the sweat simulation study, along with direct headspace analysis. So a little bit of a break from convention with regards to leachables testing, but really with the logistics of this patch and what was available to us, this was really our only option. And so we developed those methods for the target analytes, including that 2,6-xylidine, but we also made sure that the methods were sufficient for general screening of any unknown leachables that may develop. We moved into method validation. The headspace method was validated for pinene, terpenine, dioxylane, methylhexane 3-ene, and methylethyl ketone. GCMS was validated for cyclomethicone, DHT, hexane diol diacrylate, heptadecane. LCMS method, dibutylamine, dimethyl adipate, 2,6-xylidine, antioxidant 1098, 2-butoxy-4 dimethylaminobenzoate, and a thiophene compound. And then the ICP-MS was validated for the ICHQ3D 24 target metals. So, and again, these compounds came specifically from our extractables data, which was then assessed by the toxicologist. Once we had those methods validated, we then set up our leachables testing. This was a leachable study where we took the patches, we put them into both room temperature and accelerated storage. The room temperature storage was out to 24 months with samples taken at 0, 3, 6, 12, 18, and 24 months. And then the accelerated storage samples were taken at 0, 1, 3, and 6 months and analyzed for leachables using those validated methods. The patches were stored in their pouches, so that way we could test and monitor for any leachables coming specifically from the pouch, as well as the patch materials. And again, we're always screening for both our target leachables as well as any unknown leachables. And then we use permissible daily exposure, PDA data, for any generated leachables detected above the thresholds. And that concludes my talk today. Again, thank you for your time. Thank you for your attention. And thank you to MassMedic for hosting this webinar and for asking me to speak. With that, I will stop my screen share and I'll look for any questions that we have. All right. Looks like I've got 3 questions. All right, so the first question, what is the application of this system to delta V and Yokogawa control system. I apologize. I don't know what those two references are to. I'm not really sure what those are. I can't answer that question. I've actually not come in contact with those two things. So I'm not able to answer that. I'll move on to the next one. So what are the necessary tools and standard used during testing principles? So, really the tools, as I kind of outlined, just in a general sense, is certainly a knowledge of the regulations, knowledge of expectations of the FDA. The materials risk assessment is a tool that we use to design our studies. And then certainly there's the tools in the laboratory as far as the various instruments that I reviewed. And then in terms of standards, there's obviously testing standards. I mentioned the USP 1663 for extractables, 1664 for leachables. And then with regards to standards, obviously we're acquiring chemical standards as needed and as I reviewed. Had a question, what is the application of this system to oil and gas industry? Really, I'm not aware of the oil and gas industry having any specific concerns about extractables and leachables. I think with regards to the oil and gas industry, if you're looking at extractables and leachables from, whether it be storage systems or maybe transportation systems of oil and gas products, that leachables would be more of a performance concern than they would be a safety concern. Obviously, oil and gas isn't being, consumed or in contact with people. And so I think your concerns there would be, do those leachables affect performance of your oil or gas product, in the form of an impurity, obviously, in your gas, could that affect performance of the gas that's used by the consumer? And so I think that would be your focus there is analyzing for those impurities that could leach out from those contact materials such that they could be, effect on performance. I think that's where you look at those limits. How do you validate methods? Do you spike patches with analytes, extract, and test? So generally, when we're validating leachables methods, we're going to look to validate for the ICH general guidance for method validation. In the case of the case study that I mentioned, again, we were doing a sweat simulation study as our test methods. So yes, we did spike patches where we could. We also spiked that sweat simulating solvent in certain instances. And then we evaluated things like accuracy, precision, intermediate precision, linearity, and solution stability, and the other main elements that go into a method validation. Aside from real-time aging, what was the time frame for your case study from start to finish? Generally, method development is going to take around a month and a half. Method development from there, once you've done your extractables testing, let me back up. Extractables testing generally takes a month and a half. Then we take that data, use that to compile our list, and then we move into method development. That's usually about a month, sometimes less. And then we move from there into method validation. That usually takes 4 to 6 weeks. And then at that point, we've got our validated methods, and then we're employing those as part of the leachables testing, which again, from there, then we're pulling samples on a monthly basis, and that testing takes usually 2 weeks to do with those validated methods. And then it looks like the last question I have, at what point are tests conducted to understand how the drug reacts with the skin? So those sorts of tests really are usually done during drug development. That would be done early on. That's where you're evaluating if the drug, not only has the desired effect and efficacy, but does it have any, toxicity or any adverse effects during administration. So that would be done very early on before you move into, further development of the patch system, packaging systems, and all of that. That would be pretty early on. It looks like I've got something in the chat. Let me look here and see.
Actually, I have a question, Eric, if you don't mind me chiming in.
Yeah.
Because I think the chat, when you answered it, was someone repeating.
Okay.
You said early on in the program, when you go through some of the processes, FDA will come back and ask for things that they've never quite asked for before, and you just say, okay, great, we'll do that. How often does that happen? And how do you think, when you're explaining this to a company who is kind of working with you and contracting with you to run the tests that are needed, how do you kind of explain those interactions and prep them for what may come that you're not expecting?
Yeah, so, the first thing I would say is you always want to try to have a working relationship with the FDA. That's not always possible. And, sometimes the FDA obviously has their concerns and they draw their lines and their limits on, how much they can really interact and try to lean back on just regulations and enforcement of those. But wherever you can have an interaction, when you've got a unique drug product or a unique packaging system, or in this case, a unique drug delivery system, I think gives you an opportunity to interact with the FDA. And I think that's going to be your best case. But again, with the interaction and when you have a unique type of product, you're going to have unexpected things that come, not only out of your testing results, but also, from the FDA as far as what their expectations are. Because with regards to a drug product delivery patch, the FDA is going to look at that as a combination product between drug product and medical device. And so there's a couple different sets of not only regulations there, but also expectations that they have to balance in their response and in their requirements of you.
And when you're engaging with companies to try to put together this testing plan, how does that work? Do you sit down with each company and kind of understand what end goals are? And as you said, come up with that methodology, come up with your list, come up with your testing plan. And then how often are you really interacting with the folks that you're working with to help kind of craft that?
Yeah, so initially there's obviously a flurry of interaction, lots of discussion. It's first defining the scope of the study, what the client's needs are. Some clients are able to do some extractables work in-house and they want to come to us for leachables. Some clients, they want us to do extractables and maybe they do leachables. And, you can imagine that there's a balance there. Whereas some clients just come and say, here, do everything. And then there's also an early interaction where I have to do some education of the client. Because they don't understand the regulations as far as extractable leachables. And as I mentioned early on, they're changing right now quite dramatically as time moves on. And so clients come and either they know nothing about extractable leachables or they have an expectation from 2 years ago, which is, not the case now. And so there's a lot of education, a lot of interaction there. But once we define that scope and, we start to get into the laboratory, that's when we'll set up a regular cadence of interaction with the client where we're sharing results as we get them, so there's no surprises. And then also we can, if we see something that's concerning, we can very quickly communicate to the client. And then if there's any changes, unfortunately, or hopefully not, that need to be made to packaging or drug product system or whatnot, that can be made in a timely fashion to hopefully avoid any delays in scheduled release of the product to market.
That's awesome. Thank you. I'm glad that you cover a little bit of that, because I think that's always been a very interesting perspective from my opinion. I come from a product development background, so I spent a lot of time with product development firms and always just understanding, as we work with startups and large companies, at what point do you have to kind of get someone else involved to help craft that plan? And at what point is an outside perspective really the best perspective you could have? Because as you said, regulations change so quickly and are updating right now. What you think is the case study is, it may be, even 6 months to a year ago, it might have been completely different. Yeah,
no, it definitely is. And so that's why, we try to stay very up on the regulations. We attend, not only we present it to the industry as we are here with MassMedic, but we also attend the various extractable leachables conferences where there's more of an interaction amongst, other, companies, laboratories, and the FDA attends as well. And we have, discussions about, what the state of the industry is and what expectations are. So that way we can stay attuned to those changes and be able to educate our clients and be a resource for them, whether it be testing or just a knowledge resource.
Perfect. Well, thank you, Eric. This was an excellent presentation, and thank you so much to you and Nicole and your entire team for being willing to do this. And as I said to everyone, presentation slides and video will be available. But Eric, thank you so much.
You're welcome. Thanks so much for hosting, and thanks to everybody for attending.
All right. Have a great day, everyone. We'll see you soon.
Bye.
Bye-bye.